Sawasdee krab. Hi, everybody. Nice to see you again this year. So today I'm going to talk about the topic of update on pharmacogenomics research and also the clinical implementation from the Asian and Southeast Asian pharmacogenomics country. So in these sessions, I'm going to update you the SEAPHARM Association's activity and also the research project. So I also update you the pharmacogenomics biomarker for severe cutaneous adverse drug reaction in Asian populations. And lastly, I will show you the current situations of the clinical implementations of pharmacogenomics for prevention of scar among Asian countries as well. So let me start from the first part. So as you may know that the SEAPHARM network has been established at 10 years ago to enable and strengthen the pharmacogenomics research among the various pharmacogenomics communities. Last year, it has been transformed from the networking to be the associations. As right now, we are the SEAPHARM associations, and the members are still be the same with the SEAPHARM networking, which is a 10 country in ASEAN community. So we have established the research project that work together as the resequencing project of 100 pharmacogenes, which is already finished. And there are ongoing projects such as the sequencing of HLA in these populations to establish the database among the ASEAN community populations, for example. So as I mentioned, a panel of 100 pharmacogenes among Southeast Asian populations was resequenced using the next generation sequencing platform under the collaborations of SEAPHARM or SEAPHARM associations. And a panel of 100 pharmacogenes can be categorized into 3 groups as drug metabolizing enzymes, drug transporters, and also the other genes such as NUDT15 and VKORC1. And in this report, present the data on the frequency of 100 pharmacogenes, and we report the frequency of the pharmacogenetics variants among ASEAN populations and compare the pharmacogenetics variations in each populations. So correspondingly, these data provide a useful resource for future research on pharmacogenomic guide dose adjustment among the ASEAN populations. So on the other hand, the pharmacogenomics of SCAR research, we try to establish the SEAPHARM SCAR, which is initiated by Thai SCAR. And in Thailand, so we also have another SCAR cohort in northeast of country. And as I know, in Asian country, we also have a big SCAR cohort, which is from Taiwan groups. They call as a T-SCAR and also I-SCAR from Japan, representatively. So from these SCAR groups, the biomarker for prevention of the carbamazepines and also the allophenols induced severe cutaneous adverse drug reactions has been discovered and well established in Asian populations. So the biomarker for carbamazepine has an ethnic specificity characteristic because the HLA-B 1502 is for Asian populations except for Japan and Koreans, because in these populations they use another biomarker such as which is the HLA-A 3101, which is the same biomarker for Caucasians. So on the other hand, for the biomarker for allophenol, which is the HLA-B 5801, show the broad spectrum and also the universal biomarker for not just only in Asians, but it's also in Caucasians as well. So by this case report from Thailand, it demonstrates that the biomarker for carbamazepine is not just only the HLA-B 1502, but also the HLA-B serotype 75, such as as HLA-B 1508, 1511, and 1521 was discovered in some populations such as in Thai, in Indonesia, in Filipinos, and also in Japanese. Therefore, the risk area in the serotype 75, such as should be considered in the clinical implementations for prevention of the scar in Asian population too. So for the other drug in aromatic anticonvulsants, HLA-B 1502 has been found a strong association with oxcarbazepines. However, for the lamotrigine, it has been studied in Thai, Han Chinese, and in Korean. And so it still be controversy for the biomarker in the lamotrigine induced scar. For the phenytoin, the HLA-B 1502 still be the controversy in these populations. However, we have the consensus study to find the biomarker as a CYP2C9, which is a primary drug metabolizing enzyme for phenytoins in Thai, Han Chinese, and also in Malay populations. So lastly, for the phenobarbital, the HLA-B 1301 showed a strong associations SMX for CYP2C19, which is a drug metabolizing enzyme for phenobarbital, has been strong association in Thai, Chinese, and also in Japanese. So last year, we also first discovered the biomarker for co-trimoxazole induced stress. The HLA-B 1301 showed a strong association with co-trimoxazole induced stress. And we also confirmed the association between HLA-B 1502 for the co-trimoxazole induced Stevens-Johnson syndrome. This is the first time to demonstrate that the biomarker for co-trimoxazole has the phenotype specificity characteristic, which is like the HLA-B 1502 for carbamazepines. So the HLA-B 1301 has been confirmed the associations by the whole genome sequencing platform in more sample size and various populations in Asian, such as Han Chinese in Taiwan, Thai, and also in Malay. And in this study, we also demonstrate that not just only for sulfamethoxazole, but its metabolite, such as 4-nitrosole, sulfamethoxazole, can be a trigger of immune system to initiate the severe cutaneous adverse drug reactions in this study. So we also confirm and demonstrate the reactions in vitro study at the Liverpool University under the supervision of Professor Munir and Professor Dean at the Liverpool University Laboratory. So surprisingly, the HLA-B 1301 show association with dapsone in Asian populations. So in the Chinese study, in Thai study, and also in Indonesian study as well. Interestingly, we first characterized the phenotype non-specificity characteristic of HLA-B 1301 in Stevens-Johnson syndrome and also dressed from dapsone, which is look like HLA-B 5801 for allopurinol. And recently, we also confirm the association with the more sample size and also demonstrate that not just only for dapsones, but it's also dapsone nitrosole, which is the metabolite of dapsone can bind with the HLA-B 1301 and present to T-cell receptor to trigger the immune system in silico models as shown in these figures. So recently, the HLA-DQ-B1-0301 has been found the association with anti-DM. This is a dipeptidylpeptidase enzyme for inhibitors in Thai and also in Japanese study. So I summarize you the table of the biomarker for severe cutaneous adverse drug reactions in Asian populations in the 2021 year. So let's move to the last part of my talk, which is the clinical implementation of pharmacogenomic screening to prevent scar in Asian populations. So these 4 biomarkers have been provided in clinical implementations worldwide, including in Asian countries as well. So by overall the pharmacogenomics testing in clinical implementations, the SEAPHARM Associations already surveyed and published the current situations in the members country. In this study, we found that just only Singapore and also Thailand have the clinical implementation for pharmacogenetic testing among the Asian countries. So according to the reimbursement status and focus on the severe cutaneous adverse drug reactions biomarker, in Asian country, we have no any country that implement the screening of HLA-B 5701 for as a reimbursement status in Asian country. However, for the HLA-B 1502 for carbamazepine screening in Asian country, in Singapore and also in Thailand, we can do the reimbursements for the screening of this biomarker. Fortunately, that in Thailand, interestingly, in Thailand, not just only for HLA-B 1502 that can do the reimbursement, but also recently the HLA-B 5801 can be a screening for our Thai population, because the HLA-B 5801 has been launched and announced by the National Health Security Office of Thailand for the screening in all patients who are going to treat it with the allopurinol with free of charge. So by summary, the SEAPHARM has been established to be the associations in 2020 or last years, and the pharmacogenetics biomarker to prevent the scar in Asian country has been discovered, such as HLA-B 1502 for carbamazepine, HLA-B 5801 for allopurinos, HLA-B 1301 for Co-trimoxazole and dapsone, HLA-DQB1 0301 for dipeptidyl peptidase enzyme for inhibitor induced bullous pemphigoids, and also CYP2C9 for phenytoins. According to the reimbursement status in Singapore, they can do the reimbursement for HLA-B 1502 as a national screening policy. But in Thailand, both HLA-B 1502 and HLA-B 5801 can be a screening and incorporate into the screening test in our country, and the patient no need to pay at all for the screen as a national screening in Thai populations. So lastly, I would like to acknowledge our contributions and support from my colleagues in Thailand and also international groups here, like the group from Liverpool University, Changkung Memorial Hospital, and also Vanderbilt University. And also on behalf of SEAPHARM, I would like to thank you very much, the organizing committee, for the invitations to present and join in SJSTEN in this year. And thank you very much.